It's my pleasure to introduce Dr. Julie Cocquet, who is an interim research director and group leader at Institut Cochin. She received her PhD from Paris Diderot in 2006 and then moved to London to do a postdoctoral fellowship in Paul Burgoyne's lab at the National Institute for Medical Research. And it was there that she started developing her interest and her research into sex chromosomes and epigenetics. And after her postdoc, she started an independent position in 2011 at Institut Cochin to continue studying the molecular regulation of sex chromosomes, the sex chromosome linked genes, and trying to understand the way that regulators of these chromosomes and genes contribute to reproductive developmental biology. And so she's made quite a number of significant contributions to the fields of epigenetics and reproductive biology, and we are really excited to be hosting her talk here today. So Julie, please take it away.
Thanks a lot, Jackson, for the very nice introduction and for the FN team to give me the opportunity to present our work today. Okay, so let me just move these. Yeah. Okay, so today I'm going to talk about DOT1-like. I'm escaping a little bit from the sex chromosome, but you will see that I will go back to it at the end of my talk. So I work in Paris, and my group studies spermatogenesis and actually chromatin dynamic and gene expression dynamic during spermatogenesis. The medical context of our talk, of our research, is male infertility, because actually, as you may know, male infertility affects 1 in 10 men in the general population, but in approximately half of the cases, the origin remains unknown, with strong evidence that it has a genetic and epigenetic environmental cause origin. As Jackson mentioned, we did a lot of work related to sex chromosome gene regulation during spermatogenesis and the role of these genes, the involvement of these genes in an intradynamic conflict that I'm not going to talk about today. And as you will see, what we are interested in can also have some impact on how deregulation in spermatogenesis can have a consequence on the progeny. So I hope I will convince you with this talk that spermatogenesis is actually a great cell differentiation process to study chromatin dynamic and gene regulation. So it is organized, for those who are not familiar with it, it is organized with three phases. Each of these phases has its own transcriptional program. So the first phase is the proliferation of spermatogonia. Then they differentiate and enter into the meiosis. And as you will know, there are two different divisions, of course, recombination of the genetic material, and then the production of haploid cells that are called spermatids. And these cells then undergo a dramatic differentiation to become spermatozoa, and also supported by a very specific genetic program. And this is this high diversity of the genetic program, the transcriptional program of germ cell differentiation has been demonstrated in 2013, where basically it was shown that across the different species, whatever the species, the organ which showed the highest transcriptomic complexity was in yellow, the testis, which of course is the site of germ cell differentiation. So the testis contains, of course, also somatic cells, but the testis is mainly formed of germ cells at different stages of differentiation. And so associated with this high transcriptional dynamic, there is also a very dynamic chromatin remodeling program with, of course, a meiosis. And so this is illustrated by a paper by Vara et al. In 2019, where you can see the high-C data, which show the difference according to the germ cell stage. And one particular event that we are very interested in is really specific to the germ cells, the male germ cell, and it happens toward the end of spermiogenesis. And actually, at this time, most of the histones will be progressively replaced by smaller, more basic proteins called protamines, and that leads to the high compaction of the sperm chromatin. And this process has been extensively studied, but still there are quite a number of things that remain unknown. What we know is that there are some specific histone modifications that are particularly abundant just before histones are removed, in particular H4 hyperacetylation. And we recently, several years ago now, we have observed, and others as well, that there is also a burst in H3K79 methylation, and I will go back to that later. And so it's throughout male germ cell differentiation that what we call the male gamete epigenome is established. And by epigenome, as this crowd will know, we, of course, mean chromatin organization, and of course, a few histone and histone PTM that are retained in the sperm, DNA methylation, but also the content of non-coding RNA of the sperm in non-coding RNA, which can be transmitted to the next generation. For some of them at least. And of course, if spermatogenesis is abnormal, it leads to male infertility. It can also contribute to a deregulation of the sperm epigenome, and this can be caused by genetic defect, but also toxic exposure. And potentially some of these epigenetic abnormalities can be transmitted to the progeny, whether by natural fertility, fecundation, or by assisted reproductive technologies, if, of course, we are talking about male, in a case of male infertility. Okay, so what we are studying is very fundamental because we want to understand what controls this dramatic event of chromatin remodeling and gene expression dynamic throughout spermatogenesis with a focus on the last step called spermiogenesis. And so this story today is about H3K79 methylation. So H3K79 methylation is actually quite handy to study because there's only one enzyme which is able to write this modification, and this is DOT1-like. So DOT1-like is the only K79 methyl transferase, and it is ubiquitously expressed, but particularly high in the testes and actually in male germ cells. And at not any stage, but at the stage where cells leave the meiosis and enter into the last phase of spermatogenesis, which consists in the differentiation of spermatids into spermatozoa. And so just before the histone removal and replacement by protamines. And at this stage, as I told you, there is a burst in K79 methylation. So we investigated the role of DOT1-like and K79 methylation during spermatogenesis by a classic approach, by producing a conditional knockout of DOT1-like in male germ cell, in postnatal male germ cells. And I will present the phenotypic result that we obtained very briefly because this was published in 2023 already. But what we found is that the spermatozoa of these males are abnormal. As you can see here, even if you're not familiar with the structure, you can see that the flagellum here is abnormal, and also that the sperm head of the mouse, which is characteristic on the left, on the right, you can see that it does not shape the same way. These flagella defects lead to a decrease in motility, and overall, these sperm defects lead to a reduced in vivo and in vitro fertilization, as you can see here in this graph. So then we wanted to investigate the molecular mechanism of these defects. So we focused on the last phase of spermatogenesis. So we collected spermatozoa from these males and to undertake molecular analysis. So first we did an observation by electron microscopy of the compaction of their head, and you can see here on the left the control panel shows very dark and small head of the sperm, while the knockout panel shows bigger and actually lighter with gray patches in the head, indicating less compact chromatin. By Western blot still on spermatozoa, we investigated the chromatin content of these cells, and we observed that there is a higher retention of histone detecting either histone H3, but also TH2B, which is the testis-specific H2B isoform. You can see a higher retention of those histones in the knockout compared to the control samples. Then we also looked at protamines, So this is performed by a very specific protocol of acid urea gels, and you can see both protamine 2 and protamine 1 in this system. And what was very clear is that there is a bigger form which corresponds to a premature form of the protamine 2, which is normally absent, which is not shown normally in the control samples, but it's present in the knockout samples. So meaning that there is histone and pre-protamine 2 retention in DOT1-like knockout sperm. Then, in collaboration with Delphine Flieger in Grenoble in France, we did LC-MSMS quantification of histone PTM on elongating spermatids. So we chose this particular time point because that's the stage during which histones are progressively replaced by protamines, and that's a critical stage for this process. So first, we confirm, that's always nice, that there is a decrease of K79 methylation, whether mono or dimethylation in the elongating spermatids of DOT1-like knockout males. But we also detected other significant changes here on H4. You can see that there is a reduction of K20 monomethyl, dimethyl, and trimethylation, but also, and really strikingly, a dramatic decrease in H4 acetylation, and particularly of the polyacetylated form on K5, 8, 12, and 16. And this was also observed by performing immunofluorescence detection using an anti-assay H4 antibody, where you can see here in green, this corresponds to the elongating spermatids in control, where the signal is much lower in the knockout males. So to summarize this first part, what we have observed is that normally while histones are, before they are removed, there is a burst of K79 methylation and histone H4 hypersatylation, then transition protein, I didn't talk about them, but then progressively incorporation of production and incorporation of P2, protamine 2, and then of protamine to mature form. But when DOT1-like is knocked, all those events are disturbed and eventually lead to reduced chromatin compaction, but also other sperm defects such as flagellum formation defects and also sperm head abnormalities, and overall reduced fertility. So the second part of my talk really focuses on the impact of DOT1-like knockout on gene expression. Because as you can see from the plethora of phenotypes that we observe, it cannot be that only the chromatin remodeling defect, the abnormal histone to protamine transition, leads to the flagellum defect and other associated defects. So we speculated that DOT1-like has an impact on gene expression regulation, and to determine or to identify really what are the consequences at the transcriptional level, we performed a detailed analysis throughout germ cell differentiation in 5 germ cell types using spiking RNA-seq analysis to be able to properly normalize our data. And so we used what I will call KIT- cells, which are undifferentiated spermatogonia, then differentiated spermatogonia, primary and secondary spermatocytes, and also round spermatids. So the first thing is that the MDS plot shows that the cell type, actually the samples, the replicates are clustered according to cell types, which makes sense. And then what we did on these analyses is first an enrichment, gene set enrichment analysis to look at the pathway that are deregulated according to the cell type. And first, what we see in the premeiotic cells, surprisingly, actually, is a defect, a deregulation of pathways linked to the flagellum assembly and sperm motility. And well, it's not surprising because that's one of the phenotypes that we observe here that I showed before. But what was surprising is to see it as so early, so before meiosis, while this phenomenon, this process, only occurs after meiosis here. Secondly, we observed that there were pathways associated to chromosome organization, chromosome condensation that are deregulated in secondary spermatocytes, so at this stage. And then we also observe that apoptotic pathways are deregulated, and here in round spermatids. And when that's a phenotype that didn't present before, but actually when we went back to looking at our testicular section, we could see that by doing a tunnel assay to detect apoptotic cell, we could see that there was a higher proportion of elongating spermatids, so this stage here, which are apoptotic in the knockout compared to the control. Okay, so next we perform differential analysis on using our RNA-seq data. And here you will see on the left, you can see an alluvial plot which shows the dynamic of gene deregulation throughout germ cell differentiation. So first, you can really see that the deregulation is increased according to the germ cell differentiation. So, and with the peak in the secondary spermatocyte stage, so just at the end of meiosis and entrance of spermiogenesis. Then you can see that you have more genes that are, so I will call them genes that are repressed by DOT1-like because they are genes that are found upregulated in the DOT1-like knockout. There are more genes that are repressed by DOT1-like than genes that are activated by DOT1-like in blue. And this is particularly striking at the secondary spermatocyte stage. And this actually came as a surprise initially when we did our first analysis, because K79 methylation, as you know, is described to be an active mark in all the somatic cells that have been studied, so far. And this is a typical enrichment profile that is observed. That is, the higher the peak, the higher the genes are expressed, the higher the K79 monodiphosphorymethylation peak. So we decided to produce ChIP-seq data from germ cells for K79 dimethylation to investigate where this mark is in germ cells. And we use 3 germ cell types, one corresponding to spermatogonia, which is called GSC. The second one corresponding to meiotic cells in primary spermatocyte, actually. And then post-meiotic cells, round spermatids. And as you can see from this graph, we have the same result, right? That K79 dimethylation is also an active mark in male germ cells. So the higher the expression, the higher the peak. So next, we intersect our RNA-seq data with these ChIP-seq data to see how is K79 methylation at the body of the genes that are deregulated in the DOT1-like knockout. And very clearly, we could distinguish 2 categories of genes based on the direction of their deregulation and also based on K79 dimethylation enrichment. So you can see that in blue, you have DOT1-like, the genes that are activated by DOT1-like in a wild-type situation, and they are enriched in K79 dimethylation, while genes that are repressed by DOT1-like, they are devoid of K79 dimethylation at their gene body. And this is illustrated here by images of the peaks by using IGV, where you can see 2 genes in red that are repressed by DOT1-like normally, and where there is no K79 methylation at their gene body, while conversely, 2 genes here, of which expression is activated by DOT1-like, they have a nice enrichment of K79 dimethylation. We also test by ChIP-qPCR what is the enrichment of K79 dimethylation at the gene body of the genes that are downregulated. And indeed, as you can see here, as expected actually, while in control, genes that are expressed have a high level of K79 dimethylation, and when in DOT1-like knockout, there is a decrease of K79 dimethylation at their gene body. So it suggests that DOT1-like can promote gene expression via K79 methylation, which is actually not very striking result in the sense that it was kind of expected. But what was really much more puzzling for us is to understand how the genes that are repressed by DOT1-like, what is the mechanism of DOT1-like repression? DOT1-like mediated repression, sorry. So to address this question, we looked at the chromatin environment of DOT1-like repressed genes. And for this, we used a Chromatin tool and used 7 marks, 7 gypsic marks to define the different chromatin states of our germ cells, so of the 3 germ cell type. So as for those who may not be familiar with this approach, it's based on the combination of the different marks. These define specific chromatin states, whether, for example, repressed copolycomb state or heterochromatin state or TSS, various TSS state. When we looked at the chromatin environment of the genes categories according to DOT1-like impact, we didn't see one major chromatin state for all of them, but what was clear from the comparison is that there are 3 chromatin states that are significantly increased in DOT1-like repressed genes compared to the not deregulated or DOT1-like induced genes category. And those 3 chromatin states are TSS bivalent, enhancer bivalent, and repressed polycomb. And the common feature of these chromatin states is the presence of H3K27 trimethylation. So meaning that DOT1-like repressed genes, but some of the genes that are repressed by DOT1-like, they are in an K27 trimethylation environment. So then to validate our in silico model, we performed spiking, cut and tag in DOT1-like knockout and control round spermatids using first K27 trimethylation. And what we found is, so here, bear with me because the color code has changed. So now in control, the blue correspond to control samples, while the red correspond to knockout samples. And you can see that while as expected, there is a peak in K27 trimethylation at the TSS of repressed genes in the control situation, this peak is decreased upon DOT1-like loss. And this is not observed in DOT1-like activated genes, for which K27 trimethylation is almost flat, whatever the condition. Conversely, by performing the same technical approach, the same experiment using an anti-K27 acetylation antibody, so what we can see is that the genes that are induced, of which expression is induced by DOT1-like, they are naturally, so in the control situation, they are in a K27 acetylation rich region, but they have a peak at their TSS, and upon DOT1-like loss, this peak is decreased, while less visible, but still existent. If you look at DOT1-like repressed genes, K27 acetylation is very, very poor at their TSS, but it increases when DOT1-like is lost. So this shows that DOT1-like has opposite effect on gene expression depending on the chromatin environment. And there is one last piece of information I'd like to share with you. Yes, exactly. One last piece of information I'd like to share with you is the fact that those genes are not randomly located on the genome in male germ cells. You can see here, this shows in red the location of repressed genes, and in blue the location of DOT1-like induced genes. And you can see that while repressed genes are almost exclusively on autosomes, DOT1-like induced genes are preferentially on the X chromosome here, and particularly visible in round spermatids. And this is to correlate with the fact that what is known in our field is the fact that the sex chromatin, so the chromatin of the sex chromosomes, is distinct, is not similar from that of autosomes in the male germline. And this is represented here, looking at different marks, where you can see in red the enrichment, the number of peaks per chromosome, like normalized with chromosome length. And you can see very clearly that sex chromosomes are enriched in K9 trimethylation, particularly in round spermatids. This is also true for K27 acetylation, particularly for the Y chromosome, while conversely, K27 trimethylation is less present on the sex chromosomes compared to autosomes. And this is here show K79 demethylation. And so the final piece of information, and this for us and for the person studying this process is a direct consequence of a process that you may not have or may have heard of, which is called meiotic sex chromosome inactivation. This is a phenomenon which occurs specifically in male germ cells at the time of meiosis, where the transcription of sex chromosome genes is completely shut down, while in autosome it's normal. It's maintained. And this is a chromatin-based mechanism, which is associated with a very specific structure, which is called the sex body, where the sex chromosome locate at the periphery of the spermatocyte nucleus, and which is enriched in specific marks such as gamma HOAX, but also H3K9 from methylation. And this particular chromatin environment persists somehow after meiosis in round spermatids, which now are haploid cells, so meaning that they either carry one X or one Y. And this structure, which is located adjacent to the chromocenter, is called the post-meiotic sex chromatin, and is also enriched in some of these modifications, specific modifications, such as K9 trimethylation. And if you remember the one of the first slide that I showed you, related to DOT1-like pattern of expression in the male germ cell, what is very clear is that this is a structure here is the post-meiotic sex chromatin. So DOT1-like is detected throughout the nucleus, but with a particular strong enrichment on this structure. So my conclusion here, I will recapitulate very quickly what I've told you is the fact that DOT1-like in male germ cells control chromatin reorganization and regulate genes that are involved in multiple aspects, multiple processes required for normal germ cell differentiation, and that we have contributed to a better understanding on how DOT1-like acts on gene expression, and that depends on the chromatin organization, the chromatin composition of the gene that it regulates. And so genes that are in an environment which is high in K79 demethylation and low in K27 trimethylation, in particular X-linked genes, upon DOT1-like loss are downregulated, and conversely, genes that are in an environment that are K27 trimethylation rich are upregulated upon DOT1-like loss. And I will already quite late, but we are now studying K79 methylation and other histone marks that are dynamics throughout spermatogenesis in the context of gene regulation, but also in the context of alternative splicing. And very importantly, I will acknowledge the people who have done the work. So Melina for the first part of the study, with also Com and Alberto here, and Laila and Manon also particularly for the second part of my presentation. I will acknowledge our collaborators and also the funding agencies. And thank you for your attention.
All right, that was absolutely great, really interesting. And the first thing that comes to my mind, I guess, seeing the localization of DOT1L at the sex body kind of made me question this question, but I'm wondering whether you can separate like an enzymatic role for DOT1L versus a non-enzymatic role based on the different functions you see at the sex chromosomes and the autosomes.
Yes, that would be great to have a mutation only on the catalytic domain, for instance, to try to decipher between the two, yes, the non-catalytic and the catalytic effect of DOT1-like. Indeed, we don't have it. Some people have done it in a cell model, and they find that there are some effects also on gene repression that are dependent of K79 methylation, and some that are not dependent of K79 methylation. And actually the story is more complex than that, because I really presented what we know of K79 methylation at the gene body, but actually other groups, and we have also observed that in male germ cells, have shown that K79 methylation is also dimethylation, is also enriched at enhancers. And for that, the impact is less clear than the gene body effect. So gene body mark is really associated with gene activation, but the mark at the enhancer is more complicated, and the impact can be both, actually. Yeah.
Sure. All right, we've got a couple questions in the Q&A. I guess first up is, have you done ChIP-seq for DOT1L itself? or do you just use K79 as
Well, we tried extensively,
yeah.
I have to say, and it didn't work. And well, when we were working on that, it was not shown in mouse cells. Actually, it was the antibody seemed only to work for ChIP-seq on the human cells. So we have not generated that. Yeah, unfortunately.
So then, next question, beautiful work. Do you expect K79 trimethylation behaves the same way as dimethylation?
Well, that's a very good question. And of course, that's something we'd like to investigate. I mean, the context for K79 trimethylation, depending on the context, it shows that it has a very similar pattern than K79 dimethylation, but for other contexts, it has a different place. It has been also shown to be associated with heterochromatin structure. So yes, that would be something to investigate, definitely. Yeah.
Right. And then another question in the Q&A, is there any crosstalk between DOT1L and K79 methylation and H4 acetylation, or a role for DOT1L in the histone to protamine transition?
Well, so I hope I have convinced you somehow that DOT1-like and DOT1-like at least, because when we lose this gene, this protein, there is definitely a defect in histone to protamine transition. The link between H4 acetylation is less direct, of course, because we see decrease of H4 hyperacetylation, of acetylation as a consequence of K79 dimethylation DOT1-like loss. Also, we have not shown a direct impact, but previous work from Dylan et al. In 2016 have found a more direct interaction between K79 methylation and H4 acetylation, yes, but not in our context.
Okay, it looks like that is all the remaining questions in the Q&A. I'm very sympathetic about the inability to ChIP-seq DOT1L. I think a lot of these chromatin regulators, just the conditions involved in ChIP don't seem to be very favorable at all, especially the fragmentation or the sonication of the chromatin. Have you tried cut and run?
And actually it's not as easy as for other models to generate tagged version, because that means using another mouse, but yeah, that would be the way to go. Yeah.
Have you tried cut and run or cut and tag for .1L?
That's well, actually now we're doing cut and tag, so
okay.
I think yes, that would be would like to try that. But I think really is the antibody to detect the mouse DOT1-like was not great in Western blot actually already, and for IF, so something to to try to optimize. Yeah.
Awesome. All right. Well, we're coming up at the end of the hour, and if there are no more last-minute questions, I would like to once again thank Julie and Apurva for 2 wonderful talks, and for everyone else in the audience, thank you for attending, and we look forward to seeing you again in 2 weeks at the next Fragile Nucleosome seminar. Thank you all, and I hope you all have a wonderful evening.
Thank you. Bye-bye.
Bye-bye.